Good morning, everyone. Someone gives me a thumbs up that you can hear me okay, that'd be good. All right, Kate. Thank you, Kate. All right, good morning. This is Jim Elser. I'm director of Flathead Lake Biological Station, and it's my pleasure to welcome everyone to this week's edition of Data and Donuts. We're in Zoom format today because we've had a bit of a few COVID cases on station this morning, and so we have decided to convene in virtual format for safety's sake. So thank you for your patience with that. Appreciate it. I'm excited to introduce today's speaker today, who's my friend and colleague, Dr. Diana Six, who is here on station with me teaching field ecology together. It's been my pleasure to do that for the last few years. Dr. Six is a world-renowned ecological entomologist and forest ecologist, and who's been at the University of Montana since 1997 in the College of Forestry and Conservation. And she's very well known for her work on bark beetles and white bark pine and blister rust and other sorts of interesting and important interactions that are affecting Western forests and similar phenomena globally as well. She has a bachelor's degree from Cal Poly Pomona and a master's and a PhD from the University of California, Riverside. And as I mentioned, she's been at the University of Montana for since 1997. She's an expert aquatic entomologist, even though she's a forest ecologist and fly angler, and also a fan of Norwegian death metal. And so those are all important things that you should know about Diana, other than that she's a brilliant scientist and a wonderful person. And she is going to talk about a little of the work that she's taken on recently in collaboration with me and with others working on aspects of the nutritional ecology of bark beetles from a stoichiometric perspective. And so with that, I'm going to turn it over to Dr. Six. Dr. Six, please take it away. I'm going to share her, get the screen sharing started. And I think we're all set.
Okay, sounds good. All right, thanks everybody for coming. I certainly didn't expect to be doing this on Zoom this morning. I've gotten really pretty happy that I get to talk to live audiences a lot, but this is life these days. So hopefully this will still be a good, interesting talk for you. So it's been a while since I spoke here. Last time I think I talked about some of the genetics with white bark pine and choice of mountain pine beetle. Today I'm going to talk about bark beetles, but I'm going to talk about them in a very different realm of their ecology, and that is how they interact with fungi. And from the title in my talk, you can see that they interact with fungi in a lot of different ways. Okay, there are antagonisms where the fungi can have really serious negative effects on the beetle's fitness. There's commensalisms where the fungi are basically just using the beetles as taxis to get from tree to tree, and they don't really have an effect on the insects. And then there are some of these beetles that have really important mutualisms where they gain some crucial benefits from collaborating, if you will, with these fungi. And I'll talk about that a bit in a moment. You can also see this other term in my title, and that is ecological stoichiometry. And so probably a good place to start is to explain what ecological stoichiometry is. Now, if you work on this station, I'm almost certain you know what this is, and you may even work on it. But I thought, at least for some of the students out there, I'd give just sort of a little brief introduction. And it's basically how the balance of energy and elements influence living systems, particularly in how certain ones may constrain development and growth. And quite often it involves tracking elements through an ecosystem, and quite often what we look at are things like carbon, nitrogen, and phosphorus. But you can also do this with any of the other elements that are crucial to life. Now, for those of you that may be new to this, I want to point out this book over here by Bob Sterner and Jim Elser. You may have heard of at least one of those people. But this field has been around for a while, but really these two kind of solidified it into a really solid field of study. And so if you haven't read this book, I would recommend it because it's pretty eye-opening and really changes your view of how you look at systems. So I think probably before I jump into some of the work that Jim and I have done, applying ecological stoichiometry to these systems, give a little background on how these bark beetles and fungi interact. And they've been known to interact for a long time. The first people that noticed that fungi were always found with bark beetles noticed this something like 120 years ago. And they came up with kind of the idea that the reason bark beetles carry fungi is to kill trees. So the only beetles they were actually studying were ones that killed trees. And it sort of made sense that maybe they're carrying these virulent pathogens. They introduce them into the trees, and that's how they overcome the trees, because they have to kill them or the trees will kill them. This whole idea really became kind of set in concrete and stated as absolute fact for decades, even though there really hadn't been a lot of testing or a lot of evidence to support it. And when I became a grad student, within about 2 or 3 weeks out there in the field, I was thinking, this is not happening. And so I spent the next several years kind of debunking this whole idea. And if you're interested in looking at the arguments, Mike Wingfield, one of the top pathologists in the world, and I published an annual review in 2011 that lists out 9 arguments that show why this is not happening. Now, if this isn't what's happening, then probably something else is. And from my graduate studies on to currently, I've been looking at how they may play a role in nutrients, because for one, in almost all of the mutualisms on this planet, they involve nutrients. And so I thought, well, this was probably a good possibility. And I'll be talking a lot about that today and how Jim and I have applied ecological stoichiometry to get a better understanding of that. But while it's really fun to work on these little interactions where, insects and fungi are doing all these cool things, I've always been interested in the bigger picture. And so I've always wanted to find a way to figure out, do these kind of symbioses actually influence the beetles in ways that are strong enough to influence how the beetles influence the ecosystem, so how much impact they have. And so I'll kind of end the story today talking about that. So I want to jump into talking a little about the first insect that Jim and I looked at, and this was the Western pine beetle. And this is a sister species to the mountain pine beetle, which is the famous one right now that killed something like 70 million hectares of pines over the last couple of decades. This is also an aggressive tree killer, but it goes after ponderosa pine. And you can look down here, you can see this kind of interaction graphic, and you can see that the beetles and the trees have a very antagonistic interaction. The beetles are trying to kill the trees are trying to kill the beetles. And then you can see here with the blue arrows, this is where the mutualisms exist. So this beetle, and most of the bark beetles that have mutualists, has 2 different mutualist fungi. One's an ascomycete, the other is a basidiomycete. And you can see the arrow here is a little thicker, and that just indicates that the basidiomycete's a little better. The beetles get bigger, they have more eggs, they live longer, they can probably fight a tree longer. But both of those fungi are perfectly adequate to support the beetles. Now, if you look up in the center picture, this is a pupa of the Western pine beetle. It's about to become an adult. And you can see it's in this chamber, it's chewed out, and there's this white stuff growing on the sides. That white stuff is basically, this is a big close-up, a really dense layer of the spores produced by these fungi. And so when this beetle becomes an adult, it will feed on the spore layer, consume all of that fungus that's in here, and that turns out to have two functions. When this beetle becomes an adult, this area here becomes a structure called a mycangium, and it's like a big pocket. It's like a big fungus suitcase. And so as the beetle browses on these spores, it'll pack those suitcases full of spores so they can carry it to the next tree, and so that their offspring will have these fungi as well. Something else that's really cool about these mycangia is they're not just pockets. They have glands in there that produce antibiotics that keep out everything else except those mutualists, and also grow those fungi, nurture them during kind of transport to the tree. Something else that's important is when these are adults, they have to feed on these spores in order to reproduce. We've shown if they don't produce eggs. And we think that's because they need ergosterol to produce a nucleus for producing their sex hormones. So they're really dependent on these fungi, but they're also dependent as larvae, because as larvae, these fungi are somehow really important at getting those insects the nutrients they need. Okay, we've also looked at their evolution, and these are really ancient. These have been together for a very long time. Okay, so let's jump into the work we did trying to apply ecological stoichiometry to these systems. And to do that, to start, I want to give you a little bit of an idea of how these beetles develop in a tree. So Western pine beetle adults will mass attack a tree, and the beetles bore in, and they will bore into this layer, if I can get the cursor over here, right here under the outer bark. It's called phloem. It's a living tissue, and it's the tissue in the tree that actually transports the sugars and so forth. So there's actually some nutrients there, even though it's fairly dilute. There's also some amino acids, so it's not a terrible food source. So this is where the adults burrow into. They make these tunnels through the phloem. Here's an example of what they look like, kind of scored on the sapwood, but realize they're actually in the phloem. Now, if they were kind of a normal bark beetle, they would just stay in the phloem and they would complete their entire development there. But this is a really strange beetle. It has a bizarre behavior. And what happens is once those eggs have hatched in the phloem, instead of the larvae feeding in the phloem, they tunnel immediately out into the outer bark and they develop out there. And that, you can't find a worse place to make a living in a tree than outer bark. It's dry, it's dead, it's nearly 100% lignin and cellulose, which is indigestible. So it's really a nutritional desert. So, you know, there's a lot of curiosity. It's like, how can they feed out there and actually survive? So something else going on here is that you can see this side of the cookie is not stained. It just looks like normal wood. If you look up here, these are the actual trees we sampled. Just looks like regular wood. These areas that have no staining are completely colonized by the mutualists once those adults bring it into the tree. The mutualists are just all through the sapwood and in the phloem. But you'll also see there's these really dark black patches. And an interesting thing about these dark areas is if the beetles are unlucky enough to have their eggs, oops, I didn't mean to do that. Why am I not advancing? There we go. Sorry about that. But it's really interesting. If the eggs are laid in this area here, the larvae will bore into the bark, but they die. And we don't know why. And we know that this fungus doesn't make toxins. And I need to back up a little bit and explain this fungus. This one is not carried in mycangia. It's just carried sort of incidentally on the exoskeleton of some of the beetles. Some of the mites that ride around on the beetles carry it, but it's not really something that's consistent with the beetles, but it'll be carried in by a few, as you can see. And wherever it is, the beetles die. So we're really curious about that, especially since this fungus happens to be in the genus Ophiostoma, which is the typical group that's mutualist for the normal bark beetles that develop in phloem. Yet when you find it with Western pine beetle, the beetles die. So we wanted to include this in our work as well. So in order to, I keep hitting the wrong thing. So in order to figure out what was happening here, why do these mutualists have different effects than the antagonist? And also, how do these nutrients end up out to the beetles where they're feeding? We decided to take cores. We took cores into trees that had no fungi in them, so trees that were not affected by the beetles. And then we also took cores from trees that had the beetles, and we took cores where the mutualists were, and then cores in those black stained areas. And for each of the cores, we divided these into different tissues to analyze separately. We had outer bark, we had phloem, we had what we called the outer sapwood, which is the part of the sapwood which is out towards the phloem, and then the inner sapwood, which would be in here by the heartwood. And we analyzed those separately for carbon, nitrogen, and phosphorus. And we also collected insects, the adults and the larvae, and looked to see what their carbon, nitrogen, and phosphorus contents were. And then with those data, we could calculate a number of things that might help us understand what's going on in the system. And one of these things that we calculated was the trophic stoichiometric ratio. And this is super simple. It's just the carbon to nitrogen or carbon to phosphorus ratio in a particular tree tissue divided by that same ratio in the insect. A colleague named Mikhail Filipiak determined that a TSR that's about 4 or bigger indicates there's the beginnings of a substantial mismatch between those nutrients in the tree and in the beetles' bodies. And so the bigger the TSR, the more of a mismatch. And so that just indicates that those concentrations are more dilute. And so with a bigger number, it's going to be harder for an insect to get the things that it needs to develop. And so we did this for our beetles and tree tissues, and this is what we found. And I'll just walk you through this table, and we're focusing first on the bark because after all, this is where these particular insects develop. And so if we look at what's in control bark relative to the larvae, we can see that for nitrogen, we have a pretty big mismatch. And then if we look at the antagonist bark, we see that it's pretty much the same as the control. But if we look at bark associated with the mutualist, that mismatch is still pretty big, but it's been alleviated to a degree. It's not quite as bad. And we see the same pattern for the adults, okay? The controls and the antagonist are about the same and a pretty big mismatch. But if you throw the mutualist in, it alleviates that mismatch for nitrogen. So let's look over here with phosphorus. We can see again that the control and the antagonist are both about the same and it's a mismatch. But if you add the mutualist, now you have basically a perfect match. You have the same amount of phosphorus in the insect as in the outer bark, which is crazy because this is a really severe nutritional desert. And we can see the same thing when you look here for the adults, a big mismatch in phosphorus for the control and antagonist, but you add the mutualist in and you essentially don't have a problem. So what about with the phloem? We still wanted to look at that, even though the beetles don't develop there. And we see a very different pattern. We see that for the control phloem and larvae, we have a mismatch for nitrogen, but now the antagonist is not the same as the control. Okay, what we've got is both the antagonist and the mutualist are able to alleviate that mismatch substantially. And the same thing with the adult. Again, the control is not a good place to be, but if you have the antagonist or the mutualist in the phloem, it's a better place to be as far as nitrogen is concerned. And then if you look at phosphorus, this is amazing because it means that whether you have a fungus or not, phosphorus is either a really good match or it's in excess in this case. So this kind of makes you wonder, if you're a bark beetle, why would you ever leave phloem? Okay, especially if you have a fungus, to move into something like outer bark, which is such a difficult place to live. Well, we don't really know, but my guess is, whoops, this is very touchy. My guess is that, oh yes, good one. My guess is it's just basically competition, because once these beetles have killed a tree, there's an amazing amount of other bark beetles and other insects that just kind of pile in and start taking advantage of phloem. And so maybe some of these moved out to bark to escape that competition. Okay, oops, use these. So there are other things we can look at as well. So one thing we can look for is how much of a change might you expect in these tissues if they're colonized by fungi? And again, this is super easy. You just take the carbon to nitrogen or carbon to phosphorus ratio in the control tissue, and then you subtract that same one for a colonized tissue. And so if you find a 0, that means that the fungi are really having no impact. If you have a negative value, it means that the nitrogen or phosphorus went down. And if it's positive, it means that they went up. Okay. So what did we find? Well, when you compare control to mutualist bark, we get 47.7. So this is an increase in nitrogen if you have the mutualist present. Okay. For the same kind of a comparison with the antagonist, you actually see a slight decrease, or this is a pretty small number, so this may even indicate no change. If we look at control phloem relative to mutualist phloem and with the antagonist, you see that there's an increase in nitrogen, and it doesn't matter which of those fungi are present. Then if you look at the control outer sapwood, the one that is right up next to the phloem, and you compare that with the mutualist outer sapwood, you can see that when you have the mutualist present, you get this big drop in nitrogen. And with the antagonist, you get an even bigger one. But oddly enough, when you go to the center of the tree, the nitrogen goes up if you have the mutualist, and it also goes up with the antagonist. What happens with phosphorus? Well, this is a pretty interesting story. If you look at bark and you compare the control bark to the mutualist bark, look at that increase in phosphorus. It's huge, okay? So when you have the mutualist there, the phosphorus just really cranks up. But if you have the antagonist, it eases up a little bit, or again, this is a tiny value. This may be even relatively, small or no change. If we look at phosphorus in the phloem, again, with the mutualist, it goes up with the antagonist, but nowhere near as much as in the bark. And then there's a really interesting story going on in the sapwood. If you look at the outer sapwood with the mutualist, you have this huge drop, and you have a pretty sizable one with the antagonist. I mean, after all, if things go up here, they've got to come from somewhere, right? And it looks like it's coming from the sapwood. And then if we go even deeper into the tree, with the mutualist, phosphorus becomes completely undetectable. And it also drops off quite a lot with the antagonist. And so what we've done is kind of draw this graphic so that you can kind of visualize that a little better than looking at a table. So if you look here, back to our cookie that we showed you at the beginning where the mutualist is, this is pretty much what we see happening. The nitrogen becomes depleted in the center.
Sorry.
You're putting it on the wrong side.
Oh, sorry.
There we go. So the nitrogen gets depleted in the center, and there's some moved toward the middle, and there's some moved out here to the bark. We also see the phosphorus being completely depleted in the center and being moved out to the outer bark. And this has the effect of moving these really crucial nutrients to where these insects are feeding. What's happening over here with this antagonist, though, because it appears kind of at first glance that they're doing exactly the same thing, right? Except look where the arrows end up. This fungus is really good at moving these things, but only as far as the phloem. They don't move it into the bark. And so these little beetles out here that are in the bark are next to this rich bounty of nitrogen and phosphorus, but they can't get to any of it. And so they essentially are starving to death. I actually did a study, and that's what these SEMs are from, to kind of follow these fungi around in a tree. I wanted to see where they were going and where they were accessing nutrients. And one of the really striking things I saw was that these mutualists are capable of growing out into that outer bark, and the antagonist can't. So it doesn't even have the capacity to grow there. And so it can't deliver the nutrients. So you might wonder, why would fungi do this? Why would they go in the tree and do all this work of moving nitrogen and phosphorus out to the bark? Are they doing this to be nice to the beetles, to help them? No. Fungi are not nice. They might be super cool, super interesting, but fungi are never nice. My argument would be that, like most things on this planet, nobody's doing things for other organisms. They're doing it for themselves. And these are what are considered byproduct mutualisms, okay? In byproduct mutualism, some organism creates an effect or a substance as a byproduct of what it's doing for itself, and that happens to benefit another organism. And I think here, what we're seeing is that the fungi are doing this energetically expensive thing, like elemental translocation of these elements. They're doing it for themselves. So these beetles develop out in the bark, and they are basically the Uber for these fungi. So if these fungi want to get a ride to the next tree where they can forage and reproduce again, they have to reproduce in the presence of those beetles. So they're going into the tree, they're foraging, they're getting what they need, and then they're sending nitrogen and phosphorus out to support their own reproduction, produce their own spores, so that they can get a ride on the beetle. And you end up with this really cool byproduct that at the same time, they're delivering those same nutrients to the beetles. At least that's my hypothesis. And with some of these, we know that that evolution has really fine-tuned these. And now they're specific, they're obligate, and they can't live without each other. Others not so much, as we'll talk about in a moment. Okay, there's other things we can figure out with these data. One of those is to calculate the threshold elemental ratio, or the TER. And this is basically the concentration at which limitation or control of growth shifts from one element to the other. And here we calculated that for carbon to phosphorus. And because we're thinking of mutualists, we have to think of this in the context of what effects the mutualists might be having on this relationship. And so if we get a low TER value, that kind of indicates that these mutualists are able to really alleviate this low phosphorus environment. But on the other hand, if we get a high TER, that means that even if you have really efficient mutualists, like this beetle does, because look, they completely deplete the phosphorus in the middle of the tree. They're really good at getting all of it. But if you have a high TER, this indicates that their only can still do so much, right? It's still a low P environment. And that means that you'd have to have additional physiological adaptations by the insect to live there. So to calculate our TER, we took the insect carbon to phosphorus ratio and multiplied that by the gross growth efficiency for phosphorus divided by the gross growth efficiency for carbon. And typically how you would calculate growth efficiency is to look at the change in mass per unit of whatever the substrate is that's ingested. But we can't do that with bark beetles. They grow in big trees. They're not amenable to feeding studies. And so we use some estimators. For phosphorus, we used GGE of 1. And for carbon, we used both a low and a high efficiency scenario, low efficiency of 0.5 and high of 0.8. And so what did we find? Well, this is what we found. For the larvae, for low efficiency, 6672, high efficiency, 4170. For the adult, low is 2816, and for the adult, high was 1760. So what does that mean? Well, you'll have to take my word and Jim's for it, but these are really, really high TERs, okay? So this indicates that even with really efficient mutualists that are grabbing as much phosphorus as they can and delivering the vast majority of it to the beetles, these beetles still have incredibly low amounts of phosphorus in their body. So they've had to adapt as well to live in this situation and have a much lower phosphorus demand. How low compared to other organisms? There's been a lot of work done in this area with invertebrates, and these are some data that were compiled by Angelica Gonzalez. And you can see, and I'm not going to go through this whole table, but if you just look at kind of nitrogen to phosphorus ratios, these insects are falling in the lowest bottom percent of all of these organisms, and perhaps even the lowest. So these insects have very little phosphorus, okay, even with good mutualists. And so Jim and I were like, okay, that's cool. But that's a weirdo. This beetle is really strange. I mean, it develops an outer bark of all things. So it made us wonder, you know, what's going on with typical bark beetles that live in phloem? Because after all, phloem is a way better place to live. It's not great, but it's better. And there's a whole variety of beetles that live in phloem. There's ones that kill trees, some that don't, some have mycangian specific fungi, some don't. So we wanted to take a look at some of these and get an idea of how they compared. So luckily, we have a bunch of other beetles that coexist with Western pine beetle. And so we sampled beetles, other beetle species, in the same tree, in the same nutritional background. Okay. So we have here, this is a picture of galleries of Ips pinei. This is the pine engraver. It has its own fungus. It likes to come in and colonize trees with Western pine beetle once the Western pine beetle have killed it. We have another beetle that comes in and shares the tree. It's called Ips marginatus. It also has its own fungus. This is just a picture of the Western pine beetle with the mutualist here and the antagonist here. And then we have this other one, Hylargops reticulatus. It's a weird little bark beetle. And when it goes into a Western pine beetle colonized tree, it colonizes these patches of the antagonist. And so that one was especially interesting to me. And so we did all the same things we did with Western pine beetle with these. And instead of and I almost forgot this guy. And this is kind of the star of the show, maybe. This guy. And if you're having a hard time seeing this guy, there's a reason. This little beetle is extremely tiny, okay? It's not a bark beetle. It's actually a tenebrionid. And it lives in the galleries of these other bark beetles, all of them. And it travels around in here and it feeds on bark beetle poop. And that just seemed to me to be a crazy food, okay? Not just because bark beetle poop doesn't sound so good, but if you think about it, bark beetles are in this low nutrient environment. They're probably extracting every bit of nitrogen and phosphorus they can, so their poop should be basically non-food. Yet this is what this little beetle feeds on, so we also included it. So what did we find? I'm not going to take you through all those tables and everything, but this is a general summary. For Ips pini, we found that its fungus moved nitrogen kind of moderately efficiently and phosphorus very efficiently. Ips marginatus, its fungus, moved nitrogen and phosphorus both very efficiently. And then Hylargops reticulatus, which is with that antagonist of the Western pine beetle, relative to these other fungi, moved nitrogen and phosphorus fairly poorly. Okay, they all moved it, but in various ways. So what happened when we calculated the threshold elemental ratios for these? Well, if you look at those for Ips pini and Ips marginatus, their ters are really small. Okay, these are really packed with phosphorus. These are really different than what you see with the other beetles. Okay. If you look at Hylargops reticulatus, that was developing with the antagonist of Western pine beetle, its ters are much bigger. Okay. And if you look at, I just put Western pine beetle at the bottom for comparison because those ters were so big. Okay. Look at Corticius parallelis, that little poop eater. It has incredibly high ters. Okay. I don't know of anything out there that has such large ter values. This beetle has almost no phosphorus in its body, which is pretty amazing because phosphorus is crucial for just about everything we do. So this really was really interesting because it indicates that, you know, these different fungi have different efficiencies, and it has a huge impact on the development of their host beetles. So something else that Jim has come up with is this thing called the growth rate hypothesis. And since he's sitting in, oh, I'm getting too far from the mic. I hope I don't butcher this because Jim's sitting in the room with me. But basically, it predicts that as, you know, an insect has more phosphorus, it can grow at a faster rate. It can build a bigger body in the same period of time. With these insects, we were lucky because they all have the same length of development. So they all take about a year to develop in the tree. And so if they have different body sizes and different amounts of phosphorus, that may be related to the efficiency of their fungal partners or what's in their food. And so if you look up here, these are, let me back up a little bit. So we looked at the percent phosphorus in these insects and then looked at their dry weight as a measure of their ultimate size as adults. And you can see up here, Ips marginatus is just packed full of phosphorus. And for a bark beetle, it's relatively huge. Okay. If you look down here, you can see Hylargops reticulatus, which is small, has very little phosphorus. It's down here. Here we have Corticius parallelis, that little poop eater, which basically has almost no phosphorus and almost no body. These really kind of fit that growth rate hypothesis really well. But then we have Ips pini up here. It's super full of phosphorus, but it's little. So why is that? Well, my guess is that the symbiont of that beetle is not very good at concentrating nitrogen. And so while it has a lot of phosphorus, maybe it's nitrogen limited. Hard to say. We need to take more of a look. Okay, so just some conclusions from those two studies. The fungal symbionts of both primary, which are the aggressive tree killers, and the secondary bark beetles all translocated nutrients to where the bark beetle larvae feed. But these different fungi varied in their efficiency in delivering those nutrients. And that was reflected in their threshold elemental ratio and the nitrogen to phosphorus ratios and their final adult body size. And perhaps the most important take home, that if you decide you're going to be a beetle living in wood, don't be a poop eater. Now, to end, and it looks like I still have a little bit of time, which is good. I did mention that I wanted to take kind of these studies of looking at symbioses and these interactions between these little insects and these little fungi and try to move it out onto a bigger scale to see if they actually may have some importance in driving how much these different bark beetles influence forest ecosystems. And so I'm going to try and cram a lot into a few minutes here. I went into this graphic in detail in a paper I published in Current Opinions in Insect Science in 2020. So if you want more information, that's a good place to go because there's a lot going on here, but I'll try and make it clear. So what I've done here is to divide bark beetles into 4 groups based on their behavior out there in the forest. There are the aggressive bark beetles like mountain pine beetle and western pine beetle that kill trees, they build up outbreaks in weak trees, and then they move out and continue those outbreaks in healthy trees. So they can develop really big outbreaks. And in the ecosystems where they're found, you can see down here, they have really massive effects. They're actually considered ecosystem engineers that help drive the structure and function of those forests. Then we have these aggressive, oops, I gotta stick to these others. These aggressive secondaries, they build outbreaks in stress trees, but once they run out of stress trees, their outbreaks collapse. And so their outbreaks are never as big, and their impacts out there in the forest are considered fairly moderate. There are also non-aggressive secondaries. And these beetles they really can't kill trees unless those trees are practically dying already. They go into trees that are killed by something else, fire, bark beetles, and they never build up in very large numbers. So their impacts out there are fairly low. And then there's this really strange group, the parasites. There's only a few species, and they actually go into living trees and develop their young. They leave, the tree lives. They really are very much like a parasite, like a flea, okay? And their impacts on the forest are basically pretty much nothing. And so my idea was that we know we have these different groups of beetles. Can we look at how they interact with fungi and find patterns that may account for these differences? And so part of this has to do with how the beetles and the fungi co-construct their niche, how they help each other. And the other has a lot to do with the behavior of the beetles. So if you look over here, these aggressive beetles mass attack trees. It takes literally thousands of them to kill a tree. And so when they get in there and under that bark layer in the phloem, that means they have very high densities and there's very little phloem resource to go around. For those that are aggressive secondaries, they also mass attack, but their densities are much lower. That means there's more phloem availability. For those that are secondaries that are not aggressive, their attack densities are fairly low, lots of phloem resource available. And these parasites often attack trees in individual pairs. They're just a male and a female. And so the phloem resource is basically unlimited. So how does this relate to the fungi? Well, if you're going to mass attack a tree and there's going to be thousands of you in there and very little phloem, then it probably is a good thing to have a fungus that helps you gain the nutrients you're going to need. So as far as co-construction, these beetles kill the tree, they provide access to the fungi, they provide habitat alteration that increases oxygen and lowers moisture, which allows these fungi to actually grow. And then in return, These fungi provide nutrients, and some of them actually even detoxify some of these defenses. They're very specific. So that means these beetles have high functioning symbionts they can rely on that allows them to attack in high densities and have very low dependence on the phloem. Over here, these beetles and fungi co-construct the niche in a very similar way. The fungi are sometimes specific, sometimes a mix. Their densities are much lower, and they have much more access to phloem. And so their dependency on fungi is less. Over here, with these secondaries that aren't aggressive, we don't know much. It's a big black hole in our knowledge, and probably because there's no money or interest in looking at beetles that don't kill trees. But for the few we've looked at, it looks like they carry a whole mix of fungi. They're not consistent. They're just sort of environmentally acquired. And so they don't have reliable symbionts to provide nutrients, although some may in certain circumstances. They're typically in low densities, and they probably rely mostly on phloem. And then over here, the beetles may provide access to fungi. They tend not to carry any or very few. It's probably not a good thing for a parasite to even have a fungus because it triggers defenses, and those trees are alive for their whole life cycle. And so that would be a constant battle. They have no to low diversity amounts of fungi. They're low density in the trees and completely dependent on fungi. So if you put a lot of this stuff together, it does look like, to me, and I am fungus centric and biased, that the whole reason these really aggressive beetles are able to kill so many trees is that they're able to mass attack it, and they can only do that if they have high functioning, efficient mutualists to make up for that lack of phloem because of all the competition within the tree. So this has been my kind of attempt to pull this all together, but we still have kind of this big black hole of what's really happening in this portion of the bark beetle spectrum. And with that, I've pretty much come to the end. I want to acknowledge a few things: NSF EPSCoR for funding, Flathead Lake Biostation for support, and Adam Bowman for dealing with lots of wood and bug samples in a very nice way. And Jim Elser for getting me interested in working in ecological stoichiometry. It's opened up all sorts of ways to get into these systems that I couldn't have done any other way. And so thanks, Jim. And I'm not sure if I can take questions or not.
Why don't we take them in the chat?
Okay, we'll take questions in the chat.
Yeah, let's take them in the chat. So put the questions in the chat if you have anything you'd like. I know we got students sitting in classrooms watching. So why don't we try it that way?
I've stunned everyone.
No, they're waiting. They're trying to get to the chat box. Up, some are coming. Dalton, you can relay them from the field ecology class, I think. Let's see.
Can you hear me by chance?
Oh,
Yes.
yeah, go ahead.
Okay, I'm gonna ask this question in person.
Okay.
Here, I'm gonna go ahead and ask it really loud.
Oh.
We have someone coming up.
Hello. You were talking about the two different fungal partners, and it sounded like one was mutualistic and one was antagonistic, and I did not get the names, like how the two fungi were different from each other as far as their like they sounded like two different general types of fungus, and I was just know a little bit more about that.
There's actually three. There's two mutualists. One of them is a basidiomycete, which is kind of related to the mushrooms we see out there. And then one is an ascomycete, which is more like molds and things. So they're in very different groups. And then there's also the antagonist, which is that dark stain. And that's an ascomycete as well. So there's actually three. Two mutualists that are good for the beetle, And then there's the antagonist that they starve to death with. And I can give you the scientific names if you want them. I just didn't throw it out there.
She said yes, that would be great.
Yeah, so the one that is the antagonist is Ophiostoma minus. The basidiomycete is Entomocorticium cobbii. And the other mutualist is Ceratocystiopsis brevicomi. And I can always write those out for you when I get back over there.
We have another question coming up, if that's okay.
Yeah.
There's also one in the chat. Do you want to?
Oh, restate how it informed this study. So I should have Jim do that because I'm going to get myself in trouble. So it helps us track the nutrients in the system. And so you look at what's in the beetle, and then you know what the beetle needs, right? And then you got to figure out where it's coming from and how it meets those needs. And so that's what we were able to do here is track, okay, there's phosphorus in the tree. How is it ending up in the beetle? Okay. And it helps you know how much of a mismatch is between the tree without the fungi and then when the fungi are added in.
Yeah, I mean, one thing I would just jump in and say, one thing that ecological stoichiometry inspires us to do is to measure the elemental requirements of different organisms instead of just assuming that everything is made of carbon or energy or some single substance, right? So organisms are made of lots of different chemical elements and different proportions, and knowing which of those beetles is high in phosphorus and which is low is very informative, as it turns out. And then if you realize that an insect has X amount of phosphorus in its biomass and the wood has ridiculously low phosphorus content itself, or the bark, you realize that the phosphorus has to come from somewhere. It can't be pulled out of thin air, even like nitrogen can by nitrogen fixation. So it has to go somewhere. And that gives you a good clue that maybe something is transporting it from another part of the tree. And that's what seems to be going on with the beneficial fungi.
Yeah.
So it just leads you to ask interesting questions by realizing that organisms are complex mixtures of different fundamental chemical elements that have rules that have to be obeyed.
Yeah, and with bark beetles, there's thousands of species, and there's a lot of variability, and this lets us understand how they differ in their dependencies on fungi. Some that we didn't expect to be dependent, like the Ips species, the secondaries, are extremely dependent on the fungi to get their phosphorus. There's no way they could do it without them. And so it just really lets us know the differences in these systems and how these insects are able to get it. I mean, working with lots of people over the years has been funny because we finally realized they're getting nutrients from fungi. And I said, yeah, but from where? And they said, well, from the fungi. And I said, but the fungi have to get it somewhere. It's not like the fungi can just make it like magic. Yeah.
Okay, we have another one up here.
Mm-hmm.
Yeah, I was
Go
just
for it.
wondering about what are some of the adaptations that the beetles have developed in these low phosphorus in the low phosphorus areas in the bark. If you could go over there.
Well, it's their ability to actually do what they need to do with very little phosphorus in their body. And that's really kind of, I mean, phosphorus is crucial. You have to have it to make membranes, for ribosomes. You need phosphorus to grow, just about everything you do. It's part of DNA, RNA. And so for these insects, they have to be able to, even with really good mutualists, they have to be able to do all that stuff, but with very little phosphorus. And so they have had to develop some way to still pull all that off with a low phosphorus body. And with the Corticius, that little poop eater, it's really remarkable because, I mean, there's almost nothing in that insect, but it's also super tiny. Okay. So, yeah,
All right, any more?
yep. Something else some of these beetles have done, as I mentioned with the mycangia, they have actually evolved these really intricate structures to make sure they have their symbionts with them. And we've actually tried to get them to pick up other things, swap fungi, and they won't. So they have evolved these ways to ensure that they have their helpers with them because they won't survive without them. Okay, one more if it's there. If not, we'll end.
All right, I'll wrap it up.
Okay.
I'll come in and wrap it up.
All right.
All right. Thank you, Diana. I'm sure everyone wants to give a big hand, a roaring applause out there in Zoom land for that very interesting, informative talk. So we'll have Data and Donuts next week again. I don't know what the state of our COVID situation will be. Hopefully we'll be back in a better situation by next week and we'll be in person again, but stay tuned. But anyway, thanks everyone for tuning in and stay safe out there.